Welcome to another episode from the Veterinary Research Channel. Blood transfusion has become an increasingly vital and sometimes life-saving procedure in veterinary medicine, particularly for our canine companions. Much like in human medicine, the ability to store blood in blood banks for extended periods has revolutionized transfusion practices, allowing for readily available blood products for up to 42 days in both humans and dogs. This prolonged preservation is made possible by storage solutions and specific temperature controls, typically around 4 degrees Celsius, plus or minus 2 degrees. However, this seemingly beneficial storage period comes with its own set of challenges. During refrigerated storage, red blood cells undergo a series of biochemical and morphological alterations collectively known as storage lesions. These lesions can potentially lead to adverse reactions in the recipient after transfusion. Scientists categorize these changes as biochemical, biomechanical, or structural, all of which can contribute to immunomodulatory effects, thereby impacting the safety and effectiveness of the transfusion. These changes include alterations in energy metabolism, an increase in oxidative damage, impairments in the red blood cell's ability to deform, the formation of vesicles, modifications in the phospholipid structure, and changes in the sodium-potassium gradient across the cell membrane. The accumulation of these alterations in the blood storage bag can also lead to an increased rate of red blood cell destruction, known as hemolysis. Research in human transfusion medicine has indicated that certain components derived from white blood cells, such as pro-inflammatory mediators like lipid peroxidation products, can influence biological responses after transfusion. These responses could potentially contribute to undesirable transfusion-related complications, such as transfusion-mediated immunomodulation or a serious condition known as transfusion-related acute lung injury, which is a form of acute respiratory distress syndrome. Given the well-established benefits of leukoreduction, a process that removes white blood cells, in reducing the occurrence and severity of storage lesions and adverse events following transfusion in humans, it might seem logical to apply this technique universally. However, the practice of performing leukoreduction on human and canine blood units before storage is not yet a standard procedure worldwide. The recent advancements and application of Omics technologies in human transfusion medicine have significantly enhanced our understanding of the underlying mechanisms that contribute to the development and severity of storage lesions. For instance, Omics approaches have helped identify various factors beyond just the duration of storage that can affect the onset and progression of these lesions. These factors include the characteristics of the blood donor, such as their sex, age, and ethnicity in human blood storage, as well as the strategies used for processing and storing the blood units, such as leukoreduction and the use of specific storage additives. While a considerable amount of data exists on human blood storage, there is a relative scarcity of information on the application of Omics technologies in the field of veterinary transfusion medicine. Recent initiatives, such as the Omics project, have begun to address this gap by conducting comparative metabolomic studies to investigate the metabolic profiles of red blood cells from different animal species, including dogs, during refrigerated storage. These studies aim to identify both baseline and time-dependent differences in red blood cell metabolism across species as a function of storage time. While these comparative omics insights are valuable, more research is needed to build a comprehensive database that can effectively inform veterinary practice. Specifically, at the time of this study, no metabolomics investigation had thoroughly examined the impact of leukoreduction on the metabolic characteristics of stored canine red blood cells and the surrounding fluid known as the supernatant, which became the central focus of this research. The researchers in the study collected packed red blood cell units from 8 donor dogs at 2 different veterinary blood banks in Italy. For each donor, 2 packed red blood cell units were prepared from a 450 milliliter collection of whole blood using transfusion bags containing a citrate phosphate dextrose saline adenine glucose mannitol solution, a standard blood storage solution with an integrated leukoreduction filter. This allowed for the creation of 1 non-leukoreduced packed red blood cell unit and 1 leukoreduced packed red blood cell unit for each dog. These units were then stored in the blood bank at a controlled temperature of 4 degrees Celsius, plus or minus 2 degrees, for up to 42 days. Sterile samples were taken weekly from each unit for comprehensive omics analyses, which included both metabolomics and lipidomics to assess the metabolic and lipid profiles. The results of the metabolomics analysis revealed a significant effect of leukoreduction on both fresh and stored red blood cell metabolic characteristics. The non-leukoreduced packed red blood cell units were characterized by higher concentrations of free short and medium-chain fatty acids, specific carboxylic acids like pyruvate and lactate, and certain amino acids such as arginine and cysteine. Conversely, the leukoreduced packed red blood cell units exhibited higher concentrations of glycolytic metabolites, which are substances involved in the breakdown of glucose for energy, high-energy phosphate compounds like adenosine triphosphate, and antioxidant metabolites, including those in the pentose phosphate pathway and total glutathione. Further exploration of the metabolomics data, analyzed by metabolic pathway, indicated that leukoreduced packed red blood cells maintain significantly higher concentrations of glucose after the first week of storage. They also showed lower levels of triose phosphate compounds, but higher levels of phosphoglycerate during the initial 2 to 3 weeks of storage. Notably, the leukoreduced packed red blood cells had consistently lower concentrations of pyruvate and lactate throughout the entire storage period, despite having significantly higher concentrations of adenosine triphosphate, the main energy currency of the cell, during the first 3 weeks of storage. In contrast, the surrounding fluid of the non-leukoreduced red blood cells showed increased glucose consumption and lactate accumulation. This difference could be attributed to the presence of metabolically active platelets and white blood cells in the non-leukoreduced units, which are largely removed in the leukoreduced units. The researchers also observed transient increases in hexose phosphate in the first week of storage, particularly in the leukoreduced units, suggesting an activation of the pentose phosphate pathway, a crucial pathway for producing antioxidants. This was further supported by the finding of higher concentrations of reduced glutathione and lower concentrations of oxidized glutathione in leukoreduced packed red blood cells during the first 2 weeks of storage. The metabolomics analyses also showed that storage led to an accumulation of certain carboxylic acids, such as alpha-ketoglutarate and succinate, in the packed red blood cells. However, succinate primarily accumulated in the non-leukoreduced red blood cells, while its concentrations remained stable or even decreased in the leukoreduced units. Succinate is a byproduct of amino acid breakdown in cells containing mitochondria, such as the platelets and white blood cells present in non-leukoreduced units. Correspondingly, the non-leukoreduced units showed higher concentrations of sulfur-containing amino acids, 1-carbon metabolites, and arginine catabolites compared to the leukoreduced units, especially at the beginning of storage. In contrast, higher concentrations of sphingosine 1-phosphate were observed in the leukoreduced units. These early changes likely reflect the metabolic activity of the residual platelets and white blood cells in the non-leukoreduced units. Regarding fatty acid content, the researchers found higher concentrations of medium-chain saturated fatty acids in the red blood cells of leukoreduced packed red blood cell units throughout storage. On the other hand, non-leukoreduced packed red blood cells had higher concentrations of long-chain polyunsaturated fatty acids, particularly arachidonic acid, during the first 2 weeks of storage. This was accompanied by storage-dependent increases in arachidonic acid oxidation products in both types of units. Notably, there were significant differences in the accumulation of free fatty acids and acylcarnitines in the supernatants between the leukoreduced and non-leukoreduced units. Microbiological testing of all samples throughout the storage period yielded negative results, indicating no bacterial contamination. The findings of this study provide significant molecular evidence supporting the substantial impact of leukoreduction on the metabolic profile of canine packed red blood cells during refrigerated storage. These results not only build upon previous observations in human transfusion medicine, but also pave the way for future research focused on leveraging this metabolomics data to design, develop, and test innovative storage additives specifically for canine blood units. The researchers observed slower rates of glucose consumption in the leukoreduced units, which correlated with lower concentrations of triose phosphate compounds and reduced levels of pyruvate and lactate in both the red blood cells and the surrounding fluid. Simultaneously, these units maintained significantly higher adenosine triphosphate concentrations, at least during the initial 2 weeks of storage. Conversely, the non-leukoreduced red blood cells exhibited more rapid glucose utilization and lactate accumulation. Given that mature red blood cells rely solely on glycolysis for energy production, these findings suggest that the residual white blood cells in the non-leukoreduced samples might be contributing to the increased glucose consumption. Furthermore, the results indicated an activation of the pentose phosphate pathway, the primary antioxidant defense mechanism in red blood cells, particularly in the leukoreduced packed red blood cells. Preserving these antioxidant pathways is crucial for the red blood cell's ability to withstand hemolytic stress and subsequent red blood cell destruction both within and outside of blood vessels following transfusion. This study is the first to specifically investigate the pentose phosphate pathway in canine leukoreduced and non-leukoreduced packed red blood cells during storage. The higher adenosine triphosphate concentration observed in leukoreduced units during early storage suggests that leukoreduction may mitigate storage-induced adenosine triphosphate consumption and breakdown, which is a sign of deteriorating storage quality and poor post-transfusion outcomes. Maintaining adequate adenosine triphosphate levels is essential for preserving the function of ion pumps, the phosphorylation status of membrane proteins, red blood cell deformability, cell morphology, and ultimately, post-transfusion performance in both humans and dogs. Interestingly, the study found that leukoreduction prevented the accumulation of pro-inflammatory carboxylic acids, such as succinate, which are likely produced by the mitochondria present in the residual white blood cells of non-leukoreduced units. Succinate is known to be an immunomodulatory metabolite that can contribute to inflammatory complications. The non-leukoreduced red blood cells also showed a more rapid and greater accumulation of metabolites from the breakdown of purines, such as hypoxanthine, a marker of red blood cell destruction during storage. This suggests that leukoreduction helps to reduce adenosine triphosphate breakdown and deamination, which are hallmarks of declining red blood cell quality during storage. The increased hypoxanthine levels in non-leukoreduced units were accompanied by higher concentrations of free amino acids, indicating increased mitochondrial metabolism of these amino acids and potentially greater protein breakdown. Leukoreduced units also displayed lower concentrations of free fatty acids in the supernatant, with minimal impact on the fatty acid content within the red blood cells themselves. This finding suggests reduced membrane shedding and vesiculation, which aligns with previous studies on microvesicles in leukoreduced canine blood units. While leukoreduction may not completely prevent the formation of lipid peroxidation products, it appears to mitigate the extent of morphological changes and vesicle formation by decreasing free fatty acid accumulation in the supernatant. Given the critical role of the red blood cell membrane in maintaining cellular stability, permeability, and deformability in dogs, this observation is particularly noteworthy. The non-leukoreduced units were characterized by higher concentrations of arginine catabolites, especially creatinine, which is typically used as an indicator of kidney dysfunction when measured in a living animal. Finally, the increase in sphingosine 1-phosphate observed in the leukoreduced packed red blood cells might be due to its production by an enzyme within red blood cells, suggesting a higher concentration in red blood cells compared to white blood cells. The researchers acknowledged some limitations to their study. While they included multiple dog breeds, the limited number of dogs did not allow for the identification of breed-specific characteristics. However, their statistical analysis suggested that storage duration and leukoreduction were the primary factors influencing the observed metabolic changes, indicating a less prominent effect of breed on the majority of metabolic pathways studied. Given the known influence of blood donor genetics on storage quality in humans and mice, larger studies will be necessary to fully understand the genetic factors and the potential impact of variables such as breed, sex, and age on the quality of stored canine packed red blood cells. Another significant limitation is the current lack of comprehensive omics data on canine red blood cells in both healthy and diseased states, which would provide a crucial context for interpreting these findings. The observational nature of this study also limits the ability to draw definitive conclusions about the direct clinical relevance of these findings. Future research should include more detailed assessments of the impact of redox storage lesions and red blood cell energy balance on cellular morphology, vesicle formation, and the protein composition of the cells. In conclusion, this study offers compelling molecular evidence highlighting the substantial impact of leukoreduction on the metabolic landscape of canine packed red blood cells during refrigerated storage. These findings not only expand upon existing knowledge from human studies, but also provide valuable insights for future research aimed at developing improved storage strategies for canine blood products, potentially through the design and testing of novel storage additives. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.